Welcome, life science learners, to another installment. Trusted you guys are well and you're excited about the lesson today. What are we looking at today? Well, we're looking at RNA, protein synthesis, and gene coding. To give you context as to what we did in the previous lessons, we've looked at DNA and replication. So as a follow-on to that, today we're focusing on RNA. We're going to look at how RNA translates to proteins, and we're going to look at how that protein actually is constructed and made up. So let's get straight into the lesson. So as we always start off, we always look at a concept around some interesting questions. And so today I'd like to get you to think about this before we get into the lesson. How can a piece of DNA lead to someone having dark hair? For that matter, how do genes code for characteristics? So what is it that determines whether someone is left-handed like me or has specific characteristics? And then we go back to DNA and remember that characteristics are controlled by genes. And so genes have a specific sequence that is now going to be expressed, and we refer to that process of gene expression as protein synthesis. So let's try and understand how that happens in our lesson today. So an overview of what we're looking at today is we're looking at some important terminology and definitions. We will spend some time looking at the location of RNA. Very important is trying to understand the structure of RNA, and then we will spend some time looking at the types of RNA. So let's get straight into some key words, some words that are often used in this section and sometimes easily confused. So we're going to look at RNA today, and we're going to look at different types of RNA. And so some of the types that would come across would be messenger RNA, which is sometimes referred to as mRNA. We're also going to refer to tRNA, which is transfer RNA. We're going to look at the ribosome, which is made up of ribosomal RNA. The concept of base pairing we've mentioned before. A new concept that we're going to bring in today is called a codon, and we're going to try and understand what a codon actually is. We also need to understand the context of the word anticodon and its relation to a codon. Very important today are these two important processes, which I will draw attention to when we look at protein synthesis. And these terms are good to note as an important section that we focus on: transcription and the concept of translation. I will mention the term chromatin network. Again, that's to draw back to our understanding of what the DNA is made up of in the nucleus. We're going to draw some focus to chromosomes in relation to where we are in the cell. So let's get straight into RNA. So we've looked at RNA as a comparison to DNA. Let's try and understand what the context of RNA means to a cell. So guys, we have different types of nucleic acids, and if we go back to our nucleic acids in the past, we've looked at DNA as a type of nucleic acid. Today we're focusing on RNA. So let's try and understand what RNA is, its structure, and what are the building blocks of RNA. So again, RNA stands for ribonucleic acid. It is a type of nucleic acid. It's found in the nucleus. It's found in the other parts of the cell as well. It's important that we understand the structure and what makes up RNA. Let's get into that. So firstly, the location of RNA. RNA occurs in the nucleus. It's also found in the cytoplasm. What is important about RNA is also forms part of structures such as ribosomes. So we see that RNA is essentially found in different parts of the cell, unlike DNA, which was exclusive to the nucleus, and in some instances we looked at DNA being found in the mitochondria and the chloroplast. Let's try and unpack the structure of DNA. So we know that our DNA was double-stranded. RNA consists of a single-strand molecule. We refer to it as a polymer. Again, the term poly refers to many. So it's a structure that is made up of many monomers. And with these monomers, we refer to them as nucleotides. So RNA is made up of monomers called RNA nucleotides. So this image essentially shows you a strand of RNA, which is again single-stranded, which contains a backbone with several of these nitrogenous bases. Let's try and look at the detail in further. So what is an RNA nucleotide? As I mentioned, an RNA nucleotide is the monomer of RNA. Each of these contain 4 different types of nucleotides. Each of these nucleotides consist of 3 parts, and I'll go through these. So an RNA nucleotide has a sugar molecule, it has a phosphate group, as well as a nitrogenous base. We do know that DNA has a similar structure. So if we were to illustrate that as a monomer, we would see that we have a pentose sugar. Again, it's a 5-carbon sugar, so we refer to that sugar as a ribose sugar as compared to DNA, which was a deoxyribose sugar. Very similar, it has a phosphate group and it has nitrogenous bases. Let's try and unpack that a bit further. When we talk of the nitrogenous bases, there are 4 different types of nitrogenous bases that occur in RNA. These are called adenine, guanine, cytosine, and for the first time we see something very different to DNA, which we refer to as uracil. And uracil is a nucleotide that is present only in your RNA nucleic acids. So in this image, you would see that I've illustrated RNA as a single-stranded molecule. I've indicated the presence of the different nitrogenous bases. And what is unique about RNA is that there is no thymine present. So the absence of the thymine is replaced by a base called uracil, and that is represented using the letter U. So to recap, we've got adenine, we've got guanine, cytosine, and we have uracil, which replaces thymine. And those are the 4 nitrogenous bases when we're looking at RNA. Right. It's important that we start recognizing that RNA occurs in different forms. So we have different types of RNA. Broadly speaking, there are 3 main types of RNA that are relevant to us. As an overview, let's look at them. So we have messenger RNA, which is often referred to as mRNA. We have ribosomal RNA, which is sometimes referred to as rRNA. And if you can recollect in previous lessons, we've also referred to that as being a ribosome. So rRNA is actually a ribosome. And the third type is transfer RNA, which is tRNA. And the T is very similar, or it has some resemblance to its structure, which is a T-shaped structure, which has a unique function, and we'll look at their functions in a bit. These names are often used as a classification system based on the functions of these RNA. So let's try and understand what the function of these different RNA molecules are. Getting back to these slides. So if we look at messenger RNA, the term messenger RNA refers to the ability of this strand of RNA to carry out a message or a code. So when we try and understand what DNA codes for, so DNA codes for characteristics which are present in genes. The DNA controls the functioning of the cell. In order for the DNA to be able to carry out and send out specific instructions, it has to find a method or means of being able to do that. And so it will use the messenger RNA to be able to send out messages to specific parts of the cell to carry out a specific function or to initiate the expression of a gene. So we refer to that as messenger RNA. So again, the context of messenger RNA is that messenger RNA carries special information to the factories in the cell to carry out processes like protein synthesis. The next type of RNA which we mentioned was rRNA, or the ribosome, and this structure consists of 2 units. It's got a large subunit and a smaller subunit. And often it's indicated using a simple diagram which illustrates two different sized circles that are very closely fitted together, which often looks like a small little snowman. And these are the structures that are, it's a three-dimensional structure, which is a type of RNA that is wrapped up into a three-dimensional structure that has the ability to be able to read the mRNA. So the ribosomal RNA is where the proteins are made. But the ribosome has a very important function in reading the mRNA, trying to understand the message that comes from the DNA, and then instructing it to put together what we know as an amino acid chain. And finally, the third type is transfer RNA. And in the name transfer, we refer to that as being a molecule of RNA that will transfer what we need to make proteins. And in this case, the transfer RNA is transferring amino acids which are present in the cytoplasm. These are transported to a specific site where the proteins are manufactured, and that's to the ribosome. So these three structures are very much interconnected. So again, to give you context, the messenger RNA carries the message to the ribosome. The tRNA carries the amino acids, which I've indicated here with a little green structure. So when we look at these three structures, they play a very important role in putting together the proteins. So as a point of comparison, it is important that we recognize the difference between DNA and RNA. As we compare DNAs, we know that DNA is a double-stranded molecule that is a helix. We see that RNA is a much shorter strand, but it is single-stranded. DNA consists of deoxyribose sugar. We see that in the name, deoxyribose nucleic acid. RNA consists of a ribose sugar, and hence it's called ribonucleic acid. We've just looked at the nitrogenous bases, and these nitrogenous bases that make up DNA are adenine, thymine, guanine, and cytosine, and they occur in complementary base pairing. On the other hand, when we compare that to RNA, we see that RNA consists of 4 nitrogenous bases, with the exception that we now have uracil, which replaces thymine. So we've got adenine, guanine, cytosine, and we have no thymine, which is now replaced with a uracil base. What's also important is to recognize that in a DNA molecule, that the amount of adenine and thymine are always equal in comparison to each other, and likewise, the number of guanine and cytosine bases are equal. And that comes again from the nature of the way DNA strands are complementary to each other. And so that means there will always be a proportionate number of adenine and thymine bases, likewise guanine and cytosine being proportionately equal. However, in a single-stranded molecule like RNA, we will not see the proportional equality between the two. So adenine, uracil, guanine, and cytosine, they may occur in a different number, and hence they are not in a specific ratio equal to each other. And that's purely based on RNA being single-stranded. So as an illustration, we can look at these and compare these together. It's quite a good way to compare them. We can see DNA being we can see that DNA is a double-stranded molecule here. We can see that being single-stranded here. RNA is generally a much shorter molecule. DNA is a significantly longer molecule. We mentioned the difference between DNA having a thymine nucleotide, whereas uracil being present in your RNA. We also spoke about the ribose sugar, and we compared that to a deoxyribose sugar in DNA. And so guys, this would be an important section for us to be able to recognize as part of a reflective understanding of both DNA and RNA. It often does come up as a question where you are required to understand the differences between the two. So we've looked at the structure of DNA, we've compared that to the structure of RNA, and we've tried to understand what the building blocks of RNA was. We've looked at the types of RNA as well, guys, in the segment, And you guys have kind of spent some time looking at the comparative differences. We need to take a break, and when we get back, we're going to look at the process of protein synthesis and how proteins are produced using information from the DNA. You guys have done well. You deserve a break, short break, and we'll see you guys soon.